In this short video tutorial, we are going to begin the first of a two-part series that will explore innate immune responses and introduce you to some of the basic components of the innate response. In this video, we will focus on pattern recognition and the induction of inflammatory cytokines. The innate immune response includes things like physical barriers and chemical barriers. Physical barriers are things like skin, mucosal surfaces, mucus itself, cilia, and chemical barriers can include important pathways of inflammation such as the complement cascade. However, those physical barriers and chemical barriers are not sufficient, and when they are compromised, it is critical that we develop an innate immune response mediated by leukocytes that are specialized in their ability to induce an inflammatory response. Their production of inflammatory cytokines leads to inflammation that includes vasodilation of local capillaries and cellular infiltration, and the ability of those cells, such as phagocytes or natural killer cells, to eliminate pathogens or cells that are infected with pathogens. Now, many of these cells of the innate immune response are also important in resolution of inflammatory responses. However, we will not be discussing that component of the innate immune response at this time. And it is important to remember that the innate immune response directly gives rise to the activation of dendritic cells and to the induction of an adaptive immune response, which includes the activation of T cells and B cells. In today's tutorial, we're focusing on chemical barriers induced by complement, as well as the ability of innate immune cells to recognize and become activated in the presence of pathogens. So, immediate pathogen recognition of the innate immune system can be divided into two components. One, the activation of complement, and second, activation of innate immune cells via binding of pattern recognition receptors to pathogens. And this is shown here. Complement can be activated by exposure to pathogens, and innate immune cells like macrophages that are present in compromised tissues can be activated by exposure to pathogens. In this case, this is just a skin barrier that has been compromised, leading to the introduction of bacteria into that tissue. Complement functions through three main mechanisms to limit pathogen spread. First, complement can induce lysis of pathogens. Second, complement can enhance the activity of innate immune cells by enhancing their ability to participate in phagocytosis. And lastly, complement can enhance the inflammatory response by inducing vasodilation of local capillaries. Okay. Complement is activated by pathogens directly, as shown here, or by pathogens coated by antibodies, as shown here. This leads to a proteolytic cascade in which complement begins to generate complement fragments, each with their own bioactive properties. Some complement fragments can bind covalently to the surface of pathogens, and when this happens, this enhances the ability of phagocytes like neutrophils and macrophages to recognize these pathogens via complement receptors and pull them into their phagolysosome and eat them and destroy them. For that reason, this is a process called opsonization, and sometimes these fragments are referred to as opsonins because they are able to enhance phagocytosis by these phagocytes in the surrounding area. Complement fragments are also important for inducing inflammation because they can act on local capillaries and induce their vasodilation. And lastly, some of these complement fragments can recruit additional members of the complement family that can deposit on the surface of bacteria, induce pores, and lead to lysis of the bacteria and its direct destruction. So as a reminder, complement is a group of many inactive proteases. It's found constitutively in the interstitial fluid. When they are activated by recognition of pathogen surfaces, either bound by antibodies or not, it initiates a proteolytic cascade that releases complement fragments with important bioactive properties. Activated complement components can induce lysis of pathogens. They can enhance phagocytosis by acting as opsonins, and they can induce enhanced inflammation by inducing vasodilation of local capillaries. So those are the three main functions of complement that it will be important for you to know. aside from complement, cells of the innate immune system are capable of recognizing pathogens directly due to surface expression of pattern recognition receptors. Pathogens have unique molecular structures or molecular motifs that can be recognized by pattern recognition receptors of the innate immune system. Often these structures are referred to as pathogen-associated molecular patterns, or PAMPs, and some examples of PAMPs are listed here, which can include components of bacterial cell walls, fungal sugars, and even strange RNAs or DNAs that are generated typically during viral replication within the cytoplasm. Recognition of these types of patterns can trigger the ability of innate immune cells to recognize that there is a pathogen present and an inflammatory response is needed. This is a picture of the cell surface of an innate immune cell that is expressing a variety of pattern recognition receptors that can recognize a variety of these pathogen-associated molecular patterns. However, these pattern recognition receptors are not confined just to the cell surface. Pattern recognition receptors can be found in endosomal vesicles. Pattern recognition receptors can also be found in the cytoplasm that can recognize nucleic acid structures that are typical of viral replication in that location. The most well understood family of pattern recognition receptors are toll-like receptors. And toll-like receptors are recognized by many, toll-like receptors recognize many different pathogen-associated molecular patterns. And they're listed here, both PAMPs that are present in the extracellular environment and PAMPs that are present within endosomal vesicles after phagocytosis, for example. However, I'm not really expecting you to memorize all of these different toll-like receptors and the different PAMPs that they bind to. Rather, I want to use one of these toll-like receptors, toll-like receptor 4, as an example. Toll-like receptor 4 binds to a PAMP, or a pathogen-associated molecular pattern called lipopolysaccharide. Lipopolysaccharide is expressed at high levels by some bacteria. It can be recognized either as an extracellular molecule or as a component of the bacterial cell wall or in complex with other bacterial proteins. And when lipopolysaccharide binds to toll-like receptor 4 on the surface of innate immune cells, it induces a kinase signaling cascade within those cells that in turn induces a transcriptional response that leads to the production of inflammatory cytokines. I've listed many of these inflammatory cytokines here, but again, we're not learning the specific names of these cytokines at this juncture. Pattern recognition receptors can be expressed by all leukocytes and sometimes cells that are not leukocytes, so cells not formally of the immune response. Pattern recognition receptors are germline encoded and they have very broad specificity, and they can induce inflammatory cytokines and chemokines. The result is that these inflammatory cytokines and chemokines induce activation of endothelial cells via vasodilation, which leads to an inflammatory response. So that, in a nutshell, is what I would like you to know about pattern recognition at this juncture. And we will have an opportunity now in the next lecture to learn more about how pattern recognition and the production of inflammatory cytokines in turn leads to both local and systemic inflammatory responses.